Since primary school, we've all been taught to memorize that mitochondria are the powerhouse of the cell, and chloroplasts enable photosynthesis. But did you know that these organelles, often thought of as supporting characters in the cell, were actually once free-living bacteria? Did you know they are the actual true superstars of evolution? How did they end up inside our cells, working as essential components of life itself? This is the story of the endosymbiosis theory, a revolutionary idea that explains how complex eukaryotic cells, the cells we know and love today, evolved from simpler prokaryotes, mitochondria and chloroplasts through symbiotic relationships. Lynn Margulis first proposed this groundbreaking theory in her 1967 paper on the origin of mitosing cells, laying the foundation for one of biology's most fascinating concepts. Let's clear this up. Eukaryotic cells are large, complex, have a nucleus and organelles, while prokaryotic cells are small, simple, and lack these features. Let's rewind time to billions of years ago. Earth was a very different place. Life was simple, dominated by tiny prokaryotic cells, but then something remarkable happened, a partnership so profound it changed the course of evolution forever. The endosymbiosis theory suggests that a large host cell engulfed the smaller bacterium, but instead of digesting it, the two struck a deal.
Let me tell you something! Let me tell you something!
The bacterium provided energy or nutrients, and the host offered protection. Over time, these engulfed bacteria, called protobacteria and cyanobacteria, became, guess who? Our stars, the mitochondria and chloroplasts, the powerhouses of eukaryotic cells. As discussed in Gray's 2017 review on Margulis' legacy, in the 1960s, Margulis faced skepticism, but experiments later validated her ideas. For example, Kwan-John observed amoeba forming dependency on once harmful bacteria, mirroring ancient symbiosis. Similarly, Okamoto and Inoue documented a protist engulfing algae, kickstarting a photosynthetic partnership. These modern examples echo how mitochondria and chloroplasts evolved through time. So we know that mitochondria produces energy through respiration in cells, and chloroplasts allow photosynthesis to occur in plant cells. But how do we know this isn't just a wild hypothesis? The evidence is compelling, and it actually comes from several and multiple lines of research. The first line of evidence is double membranes. Both mitochondria and chloroplasts have double membranes. The inner membrane resembles that of prokaryotes, while the outer membrane likely originated from the host cell during the engulfing process. This structural feature strongly supports the idea that these organelles were once free-living bacteria. The second line of evidence is their own DNA. Both organelles contain their own circular DNA, distinct from the linear DNA found in eukaryotic nuclei. This DNA is similar to bacterial DNA, encoding essential proteins and enzymes, a clear sign of their prokaryotic ancestry. Modern genetics also further confirms this. Sequencing revealed mitochondrial DNA is unrelated to the host nucleus, proof of their bacterial ancestry. As Margulis once said, life did not take over the globe by combat, but by networking. The third line of evidence is independent reproduction. Mitochondria and chloroplasts replicate independently of the host cell through binary fission. Hint, hint, the same process used by bacteria. This means they don't rely on the host cell to reproduce, further linking them to their bacterial origins. The fourth line of evidence is ribosomes. The ribosomes within these organelles are similar to bacterial ribosomes, not eukaryotic ones. Specifically, they are the 70S ribosomes, which are characteristics of prokaryotes, unlike the 80S ribosomes found in eukaryotic cytoplasm. These four lines of evidence clearly demonstrate that mitochondria and chloroplasts were once free-living bacteria that became integral parts of eukaryotic cells through endosymbiosis. Scientists think endosymbiosis began about 1.8 billion years ago, when ancient prokaryotes were engulfed by larger cells and became mitochondria, powering their host with respiration. Later, a second event brought in cyanobacteria, photosynthetic bacteria that dramatically changed Earth's atmosphere. Cyanobacteria's oxygen production triggered the Great Oxygenation Event about 2.3 billion years ago, making the planet suitable for complex life. These cyanobacteria eventually became the ancestors of chloroplasts, allowing plants to photosynthesize and thrive. Lynn Margulis championed the idea that mitochondria and chloroplasts came from free-living bacteria. Though her theory was first met with skepticism, genetic evidence showing mitochondria are related to alpha proteobacteria and chloroplasts to cyanobacteria proved her right and transformed our understanding of evolution. Over time, these symbiotic relationships became irreversible. Genes from mitochondria and chloroplasts were transferred to the host cell's nucleus, a process called endosymbiotic gene transfer, making these organelles completely dependent on their hosts. So why does this matter? The endosymbiosis theory isn't just about how life evolved, it's about collaboration at its finest. It reminds us that even at the cellular level, teamwork drives innovation, and sometimes revolutionizes even life itself. Thank you for joining me through this journey in evolution. Be sure to check out my references for more fascinating insights on the symbiosis.